Slight interruption there. Welcome back. I had some trouble uploading such a large file, so the file kind of cut off of the last PowerPoint. So here's part two. Not nearly as long. It's about a 15-minute file, so just finish it up and I'll see you in part two. So here comes the next slide. Okay, so back to our myelin sheath. So myelin sheath in the central nervous system formed by multiple flat processes of oligodendrocytes, not the whole cell. It can wrap up to 60 axons at once. That myelin sheath gap is present. It used to be called the node of Ranvier. There's no outer collar of perinuclear cytoplasm. Thinnest fibers are unmyelinated. They're covered by long extensions of adjacent neuroglial. White matter, regions of brain and spinal cord with dense collections of myelinated fibers, usually fiber tracts. Gray matter, just neuronal cell bodies, non-myelinated fibers. Okay, so what makes white matter white is that myelin. Here's a picture of the artist's rendition of the oligodendrocyte. And then this slide here is my mnemonic for who's making myelin sheath in what system, in the central nervous system or the peripheral nervous system. So it's COPS, C-O-P-S. So it is central nervous system, oligodendrocytes, peripheral nervous system, Schwann cells. Very simple. Hopefully you'll be able to remember that a year from now when you are taking your boards. Histology of nervous tissue. So histo is tissue, okay? So study of tissue, of nervous tissue. It's kind of a redundant name, but that's what the book says, of neuroglia. So we're looking at those oligodendrocytes, and then we have astrocytes, those star cells, remember. There's three subtypes there, protoplasmic, fibrous, and radial. And then microglial cells, Schwann cells, satellite cells, and then I added in ependymal cells, which is not on that Manser and Gatz page 3, if you are following along with that book. Astrocytes, most abundant, versatile, highly branched glial cells. So this is a test slide right here. When I bold and underline and italicize something like that, it's a good idea to remember it. They cling to neurons, synaptic endings, and capillaries. They help to form the blood-brain barrier, so the little feet of it. Astrocyte functions include support embracing neurons, playing a role in exchange between capillaries and neurons, so blood and nerve. Guide migration of young neurons. Control chemical environment around the neuron. Respond to nerve impulses and neurotransmitters, so chemistry and electricity. Influence neuronal functioning, so helps to participate in the information processing of the brain. And here is a picture. So pause at any of these pictures to kind of get a better idea of it if you like any of those. This one's kind of cool. This is a real scanning electron microscope picture of it, kind of neat looking. Protoplasmic, fibrous, and radial astrocytes. We don't really go that much into depth like this slide is, but since the book mentioned their names, I decided to kind of throw it in there just to give you some context, but I wouldn't worry too much about memorizing it. Protoplasmic astrocytes, gray matter. Fibrous astrocytes, white matter. Okay, so protoplasmic, gray, fibrous, white. Both function as mechanical and metabolic support in response to injury, and then radial astrocytes are present during the development and form a scaffolding that helps guide growing axons. And then here's a couple of pictures. I really had to search for these. Again, it's not something that's commonly talked about, but since the book mentioned it, I figured I'd throw it in there. Microglial cells, do know these. Small ovoid cells with thorny processes that touch and monitor neurons. They migrate towards injured neurons. They can transform to phagocytize or chew up and eat up microorganisms and neuronal debris. So they're phagocytic cells. They're going to chew stuff up, act as primary immune defense cells. Cool little picture of it here. And then this article here actually said these words. It's snack time for your brain cells. So that was kind of neat looking. And it had this picture here of these cells for you as well. So new images show the brain's helper cells nibbling on synapses, the connections between the neurons. So the synapses, we used to just think this was space, right? So it was just a space. How is it going to be able to chew on space? Well, it turns out it's not just space, right? and then we see in red the microglia eating a synapse. Filopodia, the green projections are sent out by the synapse to contact the microglial cells. Kind of cool. And then this is the article that was taken from, so you can read that if that's of interest to you. That is at the bottom there. You can see the citation. So the helper cells, microglia, provide support for the brain's neurons. Researchers have proposed that the microglia engulf and eat synapses as part of a brain circuit pruning process that occurs during brain development, but no one had actually seen this process take place until now, until this article here in March of 2018. So new stuff. This is what neuroscientists fantasized about for years, but nobody had ever seen it before, is what this guy named Gross, Cornelius Gross, sounds like a Harry Potter teacher, a senior scientist at the European Molecular Biology Laboratory in Rome, who led the study, said in a statement, "These findings allow us to propose a mechanism for the role of microglia in the remodeling and evolution of brain circuits during development." So I put this in here because neural pruning is an extremely important concept to the brain. And one of the going theories that I like a lot is that an autistic brain was no longer able to properly neural prune. So when you prune back a plant or a bush, you're kind of cutting it back in order for it to grow back stronger. And the brain does the same thing here. So it prunes these synapses in this way. So this was revolutionary, just 2 years ago. So this concept is revolutionary to the idea of understanding autism and brain issues. And so if we can't neurally prune, think about a plant that you didn't prune back when you should have if you've played around in a garden before. It's just kind of wiry and not really healthy looking. We prune back these bushes so they can grow back stronger. Our brain does the same thing. So super fascinating stuff. I think it's really an interesting thing to kind of go after if you want to read that research article. Ependymal cells. So that's the little cilia in that picture on theirs. It's orange is the cilia and the blue is the cell, so kind of neat looking. Range in shape from squamous to columnar. Remember your cell morphology. So maybe ciliated, that's the hairs. Cilia beat to circulate that cerebrospinal fluid. So this is going to be part of the blood-brain barrier as well. So cerebrospinal fluid is going to be made here. They line the walls of the ventricles and the central cavities of the brain and spinal column. They form permeable barrier between the cerebrospinal fluid and the cavities and the tissue fluid bathing the central nervous system cells. Kind of neat. So those ventricles that we just mentioned there, so ventricles you can see there in blue in that picture up and to the left. So we have 2 lateral ventricles, a third ventricle, and a fourth. Okay, so those lateral ventricles, sometimes that confuses students because if we have something named 3 and 4, common sense would say I want to see something named 1 and 2 before we see something named 3 and 4. So I always get that question from students, but no, that's not it. So we kind of put common sense aside, right? When we study anatomy sometimes. I'm just playing. So the lateral ventricles, there are 2 ventricles there, and then there's the third ventricle in the center, and then the fourth. Okay, so a total of 4 ventricles. And we'll get into the specifics of the ventricular system in another chapter. It's either 4 or 5, and we'll get in that in a couple weeks. And to the right there, I put that in because it talked about the central canal of the spinal cord, and that's the middle circle there of the cord. So you can see that right in the center. There's a circle. It's the central canal. This is just a cool picture, kind of showing you what someone's rendition of the ependymal cells are. Another scanning electron microscope of the ependymal cells. Kind of looks like coral to me, coral reef kind of in the ocean, but this is in cerebrospinal fluid. Kind of neat looking. Oligodendrocytes again. So repetition helps you to remember it. So branch cells, processes wrapped, central nervous system, nerve fibers, myelin sheath. Another picture of that. A couple pictures here. Myelination. This is a scanning electron microscope of an oligodendrocyte cell. It's in the center orange there, attaching to the axon nerve fiber, the light green. and kind of neat looking. And then if that's interesting to you, you can go to the citation that I put at the bottom there. That's what the picture looks like in the book, actually in the Marie book. Satellite cells surround neuronal cell bodies. Okay? Bodies in the peripheral nervous system function similar to the astrocytes of the central nervous system. So my little mnemonic for this one, so we had COPS, right? Central nervous system oligodendrocytes. Peripheral is Schwann cells, and that was for myelin sheath. But now when we're looking at our astrocytes versus satellite cells, so satellite, astro, okay, star, we have a outer space theme going on here, right? But the mnemonic here is CAPS. So central nervous system is astrocytes, peripheral nervous system is satellite cells. There's a cool picture here showing you why it's called a satellite cell. Schwann cells. This is a neat picture. Neurolemocytes, sometimes you'll see them referred to as that. Most of the time you're just going to see them as Schwann cells though. They surround all peripheral nerve fibers and form myelin sheaths and thicker nerves. So similar function to those oligodendrocytes, it's vital to regeneration of damaged peripheral nerve fibers. We saw that when we were talking about the Wallerian degeneration and how a nerve repairs, right? This is a nice table that the Nolte book has, so I put that in here for you to kind of help you study. You can look at the side on the left and quiz yourself on the side on the right and kind of go back and forth until you get it down. And note where the principal location is for each. So you can write it out as well. That's why I put some of these blank tables in here, if that helps you out. That's how I always studied. I had to just keep writing it out. Another little quiz for your brain here, so go back and see if you can fill this out and you know where the certain structure. And there you have it. That concludes chapter 1. Thank you for making it all the way to the end of this chapter. Hopefully it was a good introduction for you. The next couple slides are going to be the citation slide, and then your Nolte book has assessments, is what they call it, so quiz questions as well. So I put on a Brightspace quiz. You have to do that one to get the points. Well, you don't have to do anything, right? That's how life works, but you have to do that one if you want to get the points. So do that when you have access to that. You don't have to pay for that, obviously, because it's on your Brightspace. But the Nolte book, you have to buy the Nolte book or somehow access a code to the Nolte book in order to access their assessments. So the next couple slides, I'll show you some of those questions so you can see what I'm talking about with that. So I don't do that with every chapter, but I wanted you to see what you're missing if you're not looking over at those assessments from Nolte. So to be clear, you have 2 quizzes this week. You have your lab quiz and your lecture quiz on Brightspace, and that's where you get your points from. So make sure you're checking out the lab. It's much smaller than the lecture that I just did. So there's a video to watch for the specimens, and then a PowerPoint with me to watch, and then a Brightspace quiz. So 2 quizzes, one for lab, one for lecture, and then the next couple slides I'm going to show you here. Stay tuned just a second here. Here is the citation slide, and then these are the Nolte quiz questions here. So check yourself, see if you can answer any of these questions. So here's question 1. Go ahead and pause at any of these. Question 2. I'm just going to show them to you here. So question 3, 4, 5, and that's it. So go ahead and answer those questions. You don't get any points for that. It's not an assignment that you have to turn in. It's really just to check your knowledge. Your board, NVCE, uses Nolte as a reference, this book right here. And these are Nolte's test questions, what they want you to get out of the chapter. So that's why it's a really good idea to know these questions. All right, thank you, and I will see you in chapter 2. So text me, call me, email me, any comments, concerns, or if you just want to reach out and say hi. Thank you. See you in chapter 2.